Welcome to the deep dive. Today, we have a really clear mission for you. We are unpacking a groundbreaking 2026 study from Cell Reports Medicine.
Right, authored by Rahman and a massive team of researchers.
Yeah, and if you are joining us to understand, the absolute cutting edge of infectious disease prevention, you're definitely in the right place.
We are going to explore a radically novel strategy to prevent HIV acquisition.
And it turns out the secret to stopping this virus might not be hyping up your immune system, but actually teaching it to stay incredibly powerfully calm.
Which is entirely counterintuitive.
It really can't be overstated, right?
No, not at all. The data cited right at the top of this paper puts it into stark perspective. I mean, by the end of 2024, there were roughly 40.8 million people living with HIV worldwide.
Wow, 40.8 million.
Yeah. Now, we do have amazing tools right now, like pre-exposure prophylaxis or pre-EP.
Right, and the science there is incredible. I mean, we have drugs like lenacapavir, which you can get as a shot just twice a year. For a lot of people, that has been an absolute game changer.
It undeniably is a game changer. But the researchers point out the real-world hurdles that prevent those tools from reaching everyone.
Like the cost.
Exactly. Cost is huge.
Right.
They're expensive. Plus, they require reliable access to healthcare infrastructure. And from a purely biological standpoint, there is always this looming threat of drug resistance.
So finding a true, durable, set it and forget it preventative measure of vaccine essentially is still the holy grail of HIV research.
That's the holy grail, and that is exactly what this study is chasing.
We are going to look really closely at a trial involving female rhesus macaques, which is an animal model that closely mirrors human biology.
Very closely, yeah.
And in this study, researchers achieved an 82.8% risk reduction against a highly pathogenic virus.
Right, an over 80% reduction.
Which is wild. And they achieved those numbers using this brilliant 1-2 punch. They paired a highly specialized engineered vaccine with a slow-release intravaginal ring that delivers a drug to essentially neuter the virus.
Yeah. So to really appreciate the results we're going to talk about today, we first have to understand the mechanics of those two very different weapons.
Let's start with punch number one then.
Right.
The vaccine. It's called the Delta V1 SIV vaccine.
Right.
Now, for everyone listening, whenever you hear Delta in a scientific context like this, it usually means something has been deleted or changed.
Exactly. In this case, the researchers actually removed a piece of the virus to make the vaccine.
Which is a crucial detail.
It is. The V1 stands for variable region 1. So I want you to picture the outer envelope of the HIV or SIV virus.
Okay.
It's covered in these structural protein loops. And the V1 loop is exactly what its name suggests, highly variable. Yeah. It mutates constantly.
So it's basically a moving target.
A very fast-moving target. Evolutionarily, it acts as a decoy. When your immune system spots the virus, it focuses all its defensive attention on this big, flashy V1 loop.
And creates specific antibodies just for that loop.
Right, but it's a trap.
Let me use an image memory trick for everyone listening. It's like the virus is a bank robber wearing a giant neon pink feathered hat.
I love that analogy.
Right. So the bank guards your immune system, memorize the hat. But the next day, the robber wears a glowing green sombrero.
Exactly. The hat mutates so fast that the guard's memory is completely useless.
That captures the dynamic perfectly.
Right.
Because the immune system is distracted by the flashy decoy, it misses the underlying structure.
Right. So in this vaccine, the researchers engineered the viral envelope DNA and protein to completely delete that V1 loop.
They just ripped the neon hat off.
They ripped it off. And by removing the decoy, they forced the immune system to look closer and target the more stable, vulnerable parts of the virus underneath.
Which is a region called V2, right?
Specifically V2, yes. The V2 region doesn't change nearly as much, so antibodies against it are actually effective over time.
Man, that is just brilliant engineering. So the vaccine trains the immune system to spot the real, unchangeable weak spot of the threat.
Exactly. Which brings us to punch number 2, the drug inside the ring.
Okay, the drug. It's called SAMT 247. Now, I was looking into how this works, and this is not your standard antiviral medication.
Not at all. It operates on a really fascinating chemical mechanism. SAMT 247 is what's known as a zinc finger inhibitor.
Zinc finger inhibitor, what does that actually mean?
Well, inside the virus, there's this crucial protein called the nucleocapsid protein, or NCP7.
Okay, NCP7.
Right. And this protein's entire job is to properly fold and package the viral genetic material, the RNA, so the virus can go on to infect other cells.
Got it.
But to hold its physical shape, this protein relies on tiny zinc atoms.
Oh, so the zinc atoms are like the rigid hinges on a suitcase, keeping it closed and intact.
Precisely. SAMT 247 chemically attacks that protein, forcing it to eject its zinc atoms.
It rusts the hinges.
Rests the hinges. And when the zinc is gone, the protein completely loses its structural integrity. The suitcase pops open.
And the virus can't package its RNA correctly.
Right. So if the virus tries to replicate,
Yeah.
it just ends up producing these immature, non-infectious viral duds.
Duds. So if we go back to our castle defense analogy, the vaccine trains the castle guards to ignore the enemy's disguises and recognize their true faces.
Yep.
Meanwhile, SMT 24/7 is like a chemical agent poured into the moat that instantly melts the invaders' weapons before they even reach the castle wall.
And the way that chemical moat is delivered is just as important as the drug itself.
The delivery system.
Right. The researchers designed a highly specialized intravaginal ring, or SIVR.
Let's talk about the physical device itself because, I mean, the engineering is wild.
It really is.
It is a flexible silicone ring, and tucked inside there is a polycarbonate cassette. And the drug, SAMT 247, is suspended in a monoline paste.
Now, monoline is amazing.
Tell me about it, because when it comes into contact with natural vaginal fluids, it structurally transforms, right?
Right. It forms these microscopic nanoscale channels.
Nanochannels. And then covering this is a special membrane made of expanded polytetrafluoroethylene.
Which is usually just called EPTFE in the materials science world.
EPTFE. So this membrane acts like a very strict bouncer at a club.
That's a good way to put it.
It strictly controls the rate at which the drug diffuses out of those nanochannels and into the surrounding mucosal tissue.
Exactly. It is designed to release an average of about 1 milligram of the drug per day.
But wait, I have to stop you there.
Oh, sure.
Because I was reading the background on this, and in a previous study, they already had this exact same drug in a gel form.
They did, yeah.
You just apply the gel before exposure, and it worked great. So why go through all the trouble to engineer a complex polycarbonate silicone nanochannel ring?
Well, if we look at the reality of public health interventions, it all comes down to human behavior.
Compliance.
Exactly. The gel was incredibly effective in a controlled laboratory setting when it was applied exactly 4 hours before exposure.
Right.
But out in the real world, relying on someone to perfectly time and apply a gel before every single exposure leads to massive compliance failures.
People just forget, or the timing is off.
Right. The ring solves that by providing sustained, continuous mucosal delivery. It's a set-it-and-forget-it shield that stays in place for 4 weeks at a time.
Okay, let's do a quick checkpoint for you listening just to make sure we have this locked in. We're about 5 minutes in.
Good idea.
First, we have a vaccine that deletes the viral decoy, forcing the immune system to target the true weakness. Second, we have a ring device forming nanochannels to continuously leak a zinc-destroying drug that neuters the virus.
That's the 1-2 punch.
Think about your own daily habits. Would you rather remember to perfectly apply a gel every single time or just rely on a continuous shield? It's a huge behavioral upgrade.
It absolutely is.
Oh.
But, having a great theory and great engineering is only half the battle.
Right. We have to see how it performs under extreme pressure.
Which means looking at the testing model, the researchers used female rhesus macaques and challenged them with a virus called SIV Mac251.
Now, SIV is the simian version of HIV, but why that specific strain?
The SIV Mac251 strain is notoriously pathogenic.
Really aggressive.
Highly aggressive. And notoriously resistant to being neutralized by the immune system.
So it sets a high bar.
An incredibly high bar for any medical intervention. Furthermore, the specific macaque testing model has a very proven track record.
Oh, how so?
It accurately predicted the outcomes of human HIV clinical trials, most notably the famous RV144 trial in Thailand.
Oh, I've heard that one.
Yeah, it remains the only human trial to ever show a modest level of vaccine efficacy. Because this animal model is so predictive, when an intervention works here, the scientific community pays extremely close attention.
Okay, let's walk through the actual gauntlet these animals faced and what the paper designates as Study 4.
Right, the efficacy trial.
They took 12 macaques, gave them the Delta V1 vaccine regimen we talked about, and then inserted the NMT-247 drug ring.
Yep.
After that, they subjected them to 14 weekly low-dose intravaginal exposures to that highly aggressive SIV MAC251 virus.
14 weeks in a row.
14 weeks of direct exposure.
And the results were striking. The combined approach to the vaccine plus the drug ring reduced the risk of viral acquisition by 82.8% compared to naive, untreated controls.
82.8%.
Yeah. And after all 14 exposures, 58% of the animals, so that's 7 out of the 12, remain completely uninfected.
That is massive, but I noticed a detail in the tissue analysis that really confused me.
Okay, what was that?
When they measured the drug levels in the actual tissue, the intravaginal ring resulted in lower peak tissue concentrations of the drug compared to the old gel method.
Ah, yes.
If the ring is delivering a lower peak dose than the gel, isn't that technically a step backward? How on earth is it still achieving an 82% risk reduction?
Well, that observation highlights a fundamental shift in how we need to view drug delivery.
Okay, lay it on me.
What we are seeing is that continuous, steady delivery completely compensates for a lower peak dose.
Make that make sense for me.
Think of it this way. The gel creates a massive, thick wall of defense, but it's temporary. If the timing is off by a few hours, that wall might have vanished by the time you actually need it.
Right.
The ring, on the other hand, provides a steady, unbroken shield. It might not be as thick at its absolute peak, but it's always there.
It never drops its guard.
Exactly. And perhaps more importantly, this steady, continuous, low-level exposure interacts with the immune system in ways that a massive temporary spike just doesn't.
Okay, time for another quick recap. We put this 1-2 punch up against a brutally aggressive simian virus for 14 straight weeks.
Yep.
The result, over an 80% drop in infection risk. And we learned that having a steady, persistent shield is vastly superior to a temporary wall, even if the peak dose is lower.
Which leaves a major question for you to ponder.
Right. How does a continuous low dose of a drug actually interact with the immune system?
This is where the biological narrative gets truly elegant.
I love this part.
The researchers didn't just measure whether the animals got infected. They took rectal biopsies to look microscopically at the mucosal tissue.
So zooming way, way in.
Right. They wanted to examine the immune correlates of protection. In plain English, they wanted to know exactly what was happening at a cellular level in the tissues of the animals that successfully fended off the virus.
And they found that the drug, SAMT-247, actually augments the immunity created by the vaccine.
It fundamentally changed the landscape of the cells guarding the border.
Specifically, they saw an increase in the frequency of a cell type called a CD73 plus MDC.
Right.
That stands for myeloid dendritic cell. Can you translate that into plain English for us?
Absolutely. Think of dendritic cells as the sentinels or the border patrol of your immune system.
Okay, border patrol.
They constantly sample the environment looking for threats. Now, the CD73 plus part means this specific variant of sentinel produces a molecule called adenosine.
Adenosine.
Right. Adenosine is a potent signaling molecule that naturally fortifies the mucosal barrier and promotes tissue repair.
So it's essentially the mortar that strengthens the structural integrity of the brick wall holding the virus out.
That's a great analogy, yes.
But the real aha moment for me was looking at the other cells they found.
The IL-10 producing cells.
Yes. The continuous drug delivery also expanded populations of cells called DC-10s, as well as specific macrophages, both of which pump out a cytokine called IL-10.
Right.
Now, IL-10 is a well-known anti-inflammatory molecule.
It is.
So if I have a highly pathogenic, aggressive virus actively invading my body, shouldn't my immune system be sounding a massive alarm and launching a fiery inflammatory counterattack?
You would think so.
Why on earth is it producing cells that tell the body to calm down?
That paradox is the dark genius of HIV and SIV.
The dark genius.
Yeah. Most viruses in the world are destroyed by inflammation, but HIV thrives on it.
Wait, really?
Yes. When your body triggers a massive inflammatory response, it rushes millions of CD4 plus T cells to the site of the infection to fight the battle.
And CD4 plus T cells are the exact cells the virus needs to infect in order to survive.
It's the ultimate Trojan horse. By sounding the alarm, your body is providing the virus with an all-you-can-eat buffet of target cells.
It's basically pouring gasoline on a fire.
Exactly. But by promoting a tolerogenic, or anti-inflammatory environment driven by that IL-10 molecule, the vaccine and the ring are essentially starving the virus of its targets.
They keep the border incredibly quiet.
They do. The study also observed an increase in IL-17 producing innate lymphoid cells, specifically NKP44 plus ILCs, which further maintain the physical integrity of the gut and mucosal lining.
So to use our castle analogy again, it's not about fighting a chaotic, fiery war at the gates.
No.
It's about maintaining a totally calm, impenetrable, structurally sound wall where the virus just wanders around outside with absolutely nothing to infect.
It's immunological judo. You use the virus's own required conditions, inflammation, and chaos against it by aggressively denying those conditions.
Okay, let's do our third recap here because this is vital.
Let's do it.
The drug from the ring doesn't just poison the virus. It signals the immune system's border patrol to release adenosine to patch the walls and IL-10 to keep everything calm.
Right. It denies the virus the inflammation it desperately needs.
So ask yourself, you're listening to this, if the border is totally calm and there is no massive fiery immune response, how does the body actually kill the virus particles that do manage to slip through?
Because some will slip through.
Right. We can't just leave them wandering around inside.
No. If you aren't going to carpet bomb the area with inflammation, you need highly specific, quiet assassins.
Quiet assassins.
And that introduces a mechanism called ADCC, which stands for antibody-dependent cellular cytotoxicity.
Let's break that down because it sounds intimidating.
It does, but it's straightforward. Remember how the vaccine deleted the flashy V1 loop to expose the stable V2 region of the virus?
The neon hat versus the real face.
Exactly. Well, the body created highly specific antibodies against that V2 region.
Right.
These antibodies bind to the invading virus, essentially painting a microscopic target on its back.
Target painted.
Then, natural killer cells specifically, a highly active subset of them, come along. They see the painted target, bind to it, and quietly, precisely destroy the infected cell without ever sounding the massive inflammatory alarm.
Oh, wow. And the data clearly showed that the sustained drug delivery from the ring actually increased this targeted natural killer cell activity.
It did.
Precision assassins. But that raises another problem for me.
What's that?
If you kill an infected cell, it leaves a dead, virus-filled cellular corpse lying around in the tissue.
Yes, it does.
And if that dead cell bursts open a process called necrosis, it is going to spill toxic debris everywhere.
Which is bad.
Right, because a toxic spill triggers the exact massive inflammatory alarm we are actively trying to avoid.
Which brings us to one of the most critical concepts in this entire study.
Okay.
Epherocytosis.
Epherocytosis.
Yes. This is the biological process where specific immune cells, like monocytes and macrophages, arrive at the scene and physically engulf and clear out dead apoptotic cells.
I picture this as immunological Pac-Man.
That's exactly what it is.
You have these specialized cleanup cells whose entire job is to swallow the dead cellular debris whole, sealing it inside themselves so it doesn't rot, leak, and trigger a fire alarm.
Exactly.
But how does that connect back to the drug ring?
Well, the study found that the calm anti-inflammatory environment promoted by the IL-10 molecule actually actively supports and enhances this epherocytosis process.
Oh, wow.
It's a beautifully coordinated loop. The barrier stays calm, the natural killer cells silently assassinate the infected cells, and the Pac-Man macrophages quietly sweep up the bodies so no toxic spills occur.
That is elegant, but I have to play devil's advocate here for a second.
Go for it.
How do the researchers know this isn't just the vaccine doing all the heavy lifting? Like, how do we know for sure that the SAMT 247 drug leaking from the ring is actively causing this calm, precision environment rather than just chemically breaking the virus's zinc hinges?
That is the right question to ask. And to definitively prove that, the researchers conducted what they called Study 6.
Study 6, okay.
They took mucosal cells from different macaques that had been immunized with a translationally relevant human HIV vaccine.
So these were completely different animals using different immunogens?
Exactly. They took these mucosal cells into a Petri dish, an in vitro environment, and stimulated them with HIV viral proteins.
Basically simulating a viral attack in a dish to see how the cells react.
Exactly. And here's the clincher. When they stimulated those cells with a viral protein plus the SAMT247 drug, those specific anti-inflammatory dendritic cells, the IL-10 production, and the precision Pac-Man cleanup functions all spiked significantly compared to when the cells were stimulated with the viral protein alone.
Wait, really?
Yes.
Wow. So the drug isn't just a chemical moat that breaks the virus's zinc fingers. It is actually acting like an immunological adjuvant.
Yes.
It is actively talking to the human immune cells, telling them to stay calm and activate the quiet precision assassins.
It fundamentally reprograms the mucosal microenvironment.
Yeah.
It's a two-pronged attack from a single molecule.
Okay, time for our final recap. Let's look at the complete picture. We have a vaccine that unmasks the virus's true face.
Right.
We have a device that continuously releases a drug.
Yep.
That drug neuters the virus by stealing its zinc, but it also acts as a chemical signal.
A very strong signal.
It tells the border tissues to patch the walls, stay incredibly calm, and use quiet precision assassins to clear out any stragglers without causing the fiery inflammation the virus needs to survive.
And it does all of this consistently, day in and day out, requiring zero daily effort from the person using it.
Man, when you step back and look at what this all means.
We're looking at a future where preventing HIV and potentially other severe mucosal infections isn't just about mechanically blocking a pathogen or launching a massive immune war.
Right.
It's about using continuous release biomedical devices to fundamentally reprogram the environment of your own body, making it hostile to the virus by enforcing absolute calm.
Well, before we wrap up, I want to bring up a fascinating, almost hidden nuance in the study's data that connects this to the broader horizon of personalized medicine.
Oh, the variance data.
Yes. The researchers noted that the exact rate at which the drug was released from the rings varied quite wildly between different individual animals.
It did. Even though the rings were engineered identically, they released different amounts of drug in different macaques.
And the release rate even varied within the same animal over time.
Right. The researchers hypothesized that this variance is driven by the individual's local microenvironment.
So factors like specific vaginal pH or cervical fluid volume.
Exactly. And hormonal changes from their cycle and even their unique vaginal microbiota, the local bacteria.
All of that literally influences how the medical device physically functions.
It does.
That is mind-blowing to me. The device is actively reacting to the highly specific, localized ecosystem of the body it is inside of.
Yeah, it proves that a one-size-fits-all device is always going to interact differently with a highly individualized biological system.
It completely changes the definition of precision medicine.
We aren't just designing a drug anymore. We are designing a dynamic interface.
Which leaves you with a profound question to ponder today.
Definitely.
As science pushes further into the realm of sustained-release medical devices for healthy individuals, whether it's for HIV prevention, contraception, or hormone therapy, could the next frontier of medicine be designing interventions that don't just blindly release a steady dose?
Right. Could we engineer devices that actually adapt and respond in real time to the unique, constantly changing microbiome and microenvironment of your specific body?
The possibility that our preventative tools could become as dynamic and responsive as our own biology is the next great leap forward.
It truly is.
Because when it comes to defending your borders, sometimes the most advanced strategy isn't building a bigger wall or fighting a louder war.
Sometimes the most powerful defense is simply engineering the perfect calm.
Keep exploring, stay curious, and we will catch you on the next deep dive.